Diving into DNA, a biology project by Aaron Lim, Amber Lin, and Athene Wong. This is Sammy. In each of his cells is DNA, located in the nucleus in the form of chromosomes. Let's zoom in. Here is one of his skin cells, or keratinocytes. It's an animal cell, and in its center lies a nucleus. The nucleus contains DNA in the form of chromosomes. Tightly packed chromatin comprised of DNA wrapped around histones. Each chromosome is huge, with the biggest in humans having 249 million base pairs. So let's look at a smaller section for more detail. Chromosomes are made of chromatin and have subunits called nucleosomes, DNA wrapped around histones. One way to think about it is that chromatin is made of beads, or histone proteins, wrapped around a string of DNA. DNA takes the form of a double helix, and each one has 4 different kinds of nucleotides: adenine, thymine, cytosine, and guanine. The order of these 4 bases determines what the DNA encodes for. But let's zoom in even more to the base level of DNA. Each DNA nucleotide contains the same phosphate group and also the same sugar, which scaffold DNA and form the backbone. Their nitrogenous bases, however, are unique and bond together in between the double helix. Now this is the molecular structure of a nucleotide, and here are their nucleosides. Pyrimidines have 1 ring, and purines have 2. There are 3 different kinds of pyrimidines. From left to right, you have cytosine, thymine, and uracil. The purines shown here are adenine and guanine. They're all organic molecules consisting of carbon, nitrogen, hydrogen, and oxygen in different configurations. All 4 of these bases are found in RNA, except for thymine, which is replaced by uracil in RNA. Its lack of a methyl group makes it less stable and much more suitable for RNA, which has to be broken down more easily. Now you might be wondering, which nucleotides bond to which? Thymine and adenine are a bonded pair linked by 2 hydrogen bonds, in an anti-parallel fashion as such. With less hydrogen bonds than guanine and cytosine, adenine-thymine bonds are easier to unwind. Because of this, you often find them at origins of replication and DNA sites that open up frequently. Guanine and cytosine are a bonded pair linked by 3 hydrogen bonds, which makes them stronger and raises their melting point, useful in thermophiles or other organisms that struggle with denaturation. Sequences of these bonded nucleotides form the double helix of DNA. Bye, Sammy. I hope you enjoyed this brief overview of the structure of DNA. Thank you for watching.